So let me first thank Jan Sandlin for the organization of this meeting and for the invitation. Today, I will focus my talk on how cerebellum impacts space and directional coding. In the brain, we can find multiple types of coding. The most famous coding or cells that has been discovered are actually place cells and grid cells because their discoverers won the Nobel Prize in 2014. Indeed, in 1971, John O'Keefe and his colleague Dostoevsky discovered in the hippocampus of rodent the existence of pyramidal cells that fire depending on the location of the navigator. They proposed that the assemblies of place cells form a brain cognitive map and could be a support for the memory of space. Much later, in 2005, the group of Maybrit and Edward Moser discovered another key component of the brain navigating system in the medial entorhinal cortex, the grid cell that they propose to encode metric about the animal's location. In between, in 1984, head direction cells have been discovered by James Frank in the postsubiculum and then discovered in many other parts of the brain, including the retrosplenial and the anterodorsal nuclei of the thalamus. Today, I will focus my talk on the activity of place cell and head direction cell under cerebellar alteration and show a tetrad recording of this place cell and head direction cell in cerebellar mutant mice freely navigating in a circular arena. Importantly, what we know about the activity of this cell, place cell and head direction cell, is that they are very sensitive to multiple types of sensory information. They are sensitive, for example, or the activity of this cell depends on external object, for example. If you rotate an object in front or in a particular arena, the position or the direction of this cell can be modified. Similarly, in the dark, these cells are able to maintain the stability of their firing, relying in that case on self-motion information. However, how this cell acquires this stability in their coding during freely exploration or navigation is still not completely understood. And this is actually the main question I tackle with my group in Paris. So concerning the cerebellum, it is a structure known to generate an output signal that derives from the comparison of the actual sensory state, we call sensation, and the prediction computed by the cerebellum. I have been working on the hypothesis that this cerebellar output signal is then used by the brain navigation system and may be crucial for a stable representation of the environment during navigation. I also hypothesized that such computation might be sustained by plasticity mechanism occurring at the level of the Purkinje cell in the cerebellar cortex. So in order to manipulate this cerebellum processing, in the lab, we use two types of cerebellar mutant mice, one called L7PKCY, which present an alteration, a deficit in the induction of long-term depression at the synapse between parallel fiber and Purkinje cell, and a second model called named L7PP2B, in which there is an alteration of potentiation at the level of Purkinje cells. So what we did is we first recorded place cell activity in mice freely exploring this circular arena in the presence of an object. So we used different sensory conditions, light condition, with the presence of this object, and dark condition in which we switch off the light and we remove the object and we shuffle the order. So to make too long story short, we observed that cellular alterations impact the stability of place cell in different sensory conditions. In the L7-PKCY mice, we specifically observe a deficit of place cell in the dark and only in the dark. By contrast, in L7-PP2B, we observed instability of place cell in the light when the mice was put back to a familiar environment. So following this result, we actually question the possible pathways between the cerebellum and the hippocampus that could explain that the cerebellum seems to influence this place cell activity in the hippocampus. So far, three pathways at least have been identified. So the cerebellum is indeed anatomically connected to the navigation circuit. One of these possible pathways goes through the head direction system, through the vestibular nucleus, the nucleus prepositus, the anterodorsal thalamic nucleus, and the retrosplenial cortex before reaching the entorhinal cortex and then the hippocampus. A second pathway is a posterior parietal cortex pathway described by Giannetti and Molinari in 2002, and going through the ventrolateral and centrolateral thalamic nuclei. And more recently, in my lab, we identify a possible other pathway, more direct one, going through the medial septum and the diagonal band. So today, I will show you a new result in my lab, unpublished result, concerning the head direction system. So head direction cell activity have been recorded while the animal was freely exploring this circular arena. Each session lasted 10 minutes, and then for the analysis of the data, we computed the trajectory map, the head direction firing rate map, and we were also interested in the ability of this directionality to be maintained over time, and this is represented here below in blue. So this is the work of Mehdi Falanezad, a fantastic postdoc in my lab, who were able to record in mice in two regions simultaneously, in the ADN, the anterodorsal nuclei of the thalamus, and the retrosplenial. Again, we use the same mice as before, the one we used for place cell recording, and we use different sensory conditions, again, light or dark condition. And here we also used an additional condition called rotation condition, R3, in which the object inside the arena was rotated by 90 degrees. So here are the questions we asked. Does the cerebellum, and in particular plasticity manipulation in the cerebellar cortex, impact head direction cell activity pattern? In particular, we examine whether the cerebellar alteration may affect the HD signal generation, the anchoring to external cues, the ability to maintain their activity using self-motion information. And finally, we also examine this, if this may affect the temporal synchronization between structure or within structure containing HD cells. So first, we found that HD cell activity in both PKCY and PP2B mice are similar in proportion compared to control. However, we found that in the L7-PKCY model, that HD cells within the dark have a lower stability compared to the control. And we observed that in both structure, in the ADN as well as in the retrosplenial cortex. In the light, this stability was perfectly maintained and even better in some cases. By contrast, in the L7PP2B models, we found a lower stability in the light, here, represented here. And more interestingly, the cell did not follow the Q rotation similar to that of control. And this is something that we observed again, both in ADN and in the retrosplenia. So as a first conclusion, just before, we also recorded two other types of cell, speed cell and angular head velocity cell. And in both cases, we found an intact linear speed and intact angular head velocity coding in both structure and in both models of mice. So as a first conclusion, we showed that the head direction cell signal generation is independent of the manipulation we made in the cerebellar cortex, suggesting that this HD signal generation could rather rely on a different system, and in particular, as it is known in the literature, depend on the vestibular system. However, the ability to anchor to external cues seems to involve PP2B dependent mechanism, whereas the ability to maintain the anchoring of these HD cell on self-motion cues seems to depend on PKCY dependent mechanisms. So to try to have a global view of what we found with these mice, one important point is that when we compare what we observed with place cell and head direction cell, we observed totally convergent results. Indeed, we observed similar instability in place and directional coding. In L7PKCY, place fields were altered in the dark, and HD activity was also unstable in the dark. And in L7PP2B mice, the instability was observed in the light for place cell, and the HD cell were had present a deficit in anchoring on the object. So what these results suggest is that PKCY and PP2B cerebellar dependent computation may modify the way put on self-motion cues. In case of PKCY, here in red, it is possible that the output signal generated by the cerebellum is too noisy to be properly used by the navigation system, explaining impaired coding in the dark. On the contrary, in case of PP2B, here in blue, the computation performed by the cerebellum could provide a strong signal and put too much confidence on self-motion signal. This could not only explain why these cells, place cell and head direction cell, are not able to anchor on the object, but also explain why in the dark the stability is very strong. The stability of this cell is also very strong. Of course, this interpretation needs to be tested. So in addition to the stability, we also tested the possible role of these two cerebellar dependent mechanisms for coordinating multiple attractor network. Indeed, while ADN and retrosplenial have their own attractor network, it is still unclear how their coordination is maintained for generating a unitary representation of direction. So what we did is we looked at the correlation between all simultaneously recorded HD cells pairs And we found a significant decrease in correlation only in PKCY mice. It's here in the red square, and only in the dark. More interestingly, we then divided pair into 3 groups, looking at correlation inside the ADN, inside the retrosplenial, or between ADN and retrosplenial. And what we found is that in these mice, in L7 PKCY, the correlation is weakened only between ADN and retrosplenial cells, and only, again, only within the dark. Therefore, these results point out that the cellular PKC dependent mechanism is required to coordinate these 2 HD networks. So here we illustrate this result with 2 cells simultaneously recorded. So each time it's, for example, here cell 1 in blue recorded in ADN and cell 2 in pink recorded in the retrosplenial. What you can observe here is that this cell, so it's a change in the correlation between light and dark. So in that case, for example, in the light, the 2 cells co-fire most of the time. And this is also clear on this spike raster, but also in this cross-correlation diagram. As you can see, this correlation in the different light conditions, L1, L2, L4, and L6. And by contrast, in the dark, we observed a weakened co-firing. This is illustrated here, again on the spike raster and on this pink line here, where you see this weakened co-firing. Here is another example that is the contrary of what we observed in the 2 previous cells. So no co-firing in the light, but co-firing that becomes more frequent in the dark. And again, this is observed on the spike raster, but also in this cross-correlation diagram, again in pink, you see that it's very different in the dark compared to the light. So in addition to the instability phenotypes that we described in a place cell and head direction cell, we additionally show that in the absence of external sensory cues, so in the dark, the coordination of retrosplenial and ADN HD attractor networks require cellular PKCY dependent mechanism. Importantly, this may indicate that the unitary representation of direction may not be innate to the HD cell circuitry, but require cellular computations when relying on self-motion cues. So let me conclude this talk. So first, in addition to their role in maintaining a stable representation of planes, we have shown here that the same distinct cellular mechanisms are also required for stable direction representation. We additionally show that the role of cellular PKC-dependent mechanism is also in maintaining a subsequent temporal coordination between thalamic and retrosplenial HD cell when the external sensory drive is removed. Altogether, so this result put forward first a new role of the cerebellum for maintaining a stable and unitary thalamocortical representation of direction, and combined with the hippocampal place cell recorded from this same mice model, this result suggests that the stable representation of the environment may require intact HD circuitry. So I thank you for your attention. I also, of course, want to thank all my team members and partners, in particular, the people who made this HD work. So again, the Mehdi Falanezad, who made most of this work, he was helped by Julia Lemero, Xenjana Zenelage, Jean-Vincent, and Christelle Rochefort. and of course, our fundings, in particular, Fondation pour la Recherche Médicale, l'ANR, Sorbonne University, and the CNRS. Thank you.